I'm Cristina Di Carluccio, and I work at the Department of Chemical Science at University of Naples, Federico II. In this presentation, I will give you an overview of my PhD project, based on the study of recognition of endogenous and exogenous glycans by specific receptors, mainly involved in immunity and adhesion. Before going into details, I'll try to convince you about the importance of studying glycans. All cells are covered with a layer of glycans involved in fundamental molecular and biological processes. The variety of glycans results from different factors, including monosaccharide composition, anomeric configuration, pattern of substitution. From how they are organized, indeed glycans can be linear or branch structure and can be linked to lipids or proteins to form glycoconjugates. Among the biological roles, glycans are involved in a plethora of fundamental functions, such as signal transduction, protein folding, cell adhesion, modulation of receptor activity, as well as different pathological processes and immunoregulation. In particular, they are involved in the critical equilibrium that occurs between an activation of immune response and immune tolerance. Within this frame, some bacteria, including pathogens as Neisseria meningitis or Campylobacter jejuni, have developed the ability to cover their surfaces of specific sugars called sialic acids, mistakenly recognized as endogenous molecules and allowing certain organisms to escape innate immune response. Based on these premises, during my PhD, I studied the protein-ligand molecular binding processes within different biological events to set the basis for the design of novel therapeutic treatments against human disease. This has been provided by the combination of different techniques. Starting from the expression and purification of recombinant proteins in both prokaryotic and eukaryotic cells, I studied their molecular binding to mammalian and bacterial glycans by physicochemical analysis, protein-based and ligand-based NMR approaches, and computational methods, in order to achieve the three-dimensional model of the complex. In this approach, I divided my PhD thesis in three main areas: cyclic binding to endogenous and exogenous glycans in the context of immunity, streptococcal cyclic glycadesin binding to glycans in the context of host adhesion, and the partially related project was based on the study of monoclonal antibodies targeting Staphylococcus aureus waldeckoi cases. To provide a picture of how we manage the study of protein-ligand molecular interactions, I'm going to describe some of the results obtained on cyclic glycadesins. These serine-rich lipid glycoproteins are expressed on the cell surfaces of several streptococci, commensal in the oral microbiota, but that can become pathogenic when entered the bloodstream. The specificity of sialoglycan adherence may influence the propensity of streptococci expressing cyclic glycadesins to cause infective endocarditis. During my PhD, I studied proteins with a high selectivity and others showing broader specificity for different O glycans decorating human salivary mucins and platelets. From ligand viewpoint, saturation transfer difference NMR allows to understand if the binding occurs and to provide the epitope mapping of the ligand, so the protons of the ligand mostly recognized by the protein. This technique is usually associated to the water-logged NMR that allows to gain information on the ligand-receptor interaction mediated by water molecules. Then a combination of molecular mechanics and dynamics with the transfer NOEC NMR experiments allows to derive the so-called bioactive conformation, that is, the conformation adopted by the ligand upon binding to the protein. From receptor viewpoint, the investigation of the binding site hosting the ligand can be achieved by expressing the labeled protein, assigning the amino acid spin systems by three-dimensional NMR spectra, and performing the protein titration with the ligand of interest. The variations in the chemical shift and intensity of the cross peaks observed in the spectra will be indicative of the amino acids involved in the binding. Thus, the combination of ligand-based and protein-based NMR techniques, together with the computational studies, provides a three-dimensional model of the complex. This is a summary of my PhD results, and in particular of all the three-dimensional complexes that I obtained for each project studied. Thank you for your kind attention.